Yeah, she's on the Zoom. Back up. Great. Yeah, I'm just going to talk quickly about, nope, preclinical models. So like animal models, mouse, flies, and those, and why do we need them in order to find treatments? Before all the researchers go into more details about what they do and which models they use. I'm going to try to be quick because we're running a bit behind, but so that's what I was presenting earlier, which is all the things that we've been working on, including so the different models, so the different aspects that are not working directly on the kids, but the ways that we can work in animals and in cells in order to test therapeutics, for example. So we have the biobank donations. They're going to be the cells right here, and we're going to have Rob talking about mouse, Jenica talking about neurons made from donations, and then we also have protein models, and I'll go over it. So why do we need preclinical models or animal models? So let's say we have potential therapeutics or potential treatments. So like drug repurposing, like Rachel mentioned, ASO, gene therapy, asparagine supplementations. There's a few things that we need to figure out in order to get them in your kids before, because we don't want to try to find the right dosage for your kid, like putting them directly in them and see what happens, right? That's too risky. Nobody wants that. There's a few things that can happen in preclinical models, so in animal and cells, that can be very helpful further down the line. So um, one of them is validate outcome measures and biomarkers. So outcome measures as what we use in a clinical trial as really a quantitative or qualitative marker of NARS1 disorder and how it acts, whether it gets better or worse when you try on a new medication. And those need to be set in place before we get into a clinical trial. Biomarkers that Luke will be talking about are just like outcome measures, but there are markers in your body that are changing or that are specific to a disease and that could be changing when you give a drug. So for example, for diabetes, it would be insulin. So something that can be easily found in the body, and we can be like, okay, this is changing and this is replicable with all the kids, and that is just a measure that just black or white measure that we can look at and say this is changing with this drug or not. So there's something we can look into plasma iPSCs, which are induced pluripotent stem cells, but basically cells that we can use just out of the body to test for therapeutics, mouse model as well. So another thing we can do is define toxicity and efficacy for a drug. As I said, you don't want to just go ahead and give a drug in a kid, or especially like prescription drugs or other drugs like that, and try to figure out what's the best dosage. Something that can help is put them in animals before, and this is usually that can translate pretty well into dosage that should be used in a kid. So yeah, again, you can do that in mouse, zebrafish, which is a see-through fish. We won't be talking about, but very helpful for research. Fibroblasts, which is the kind of cells that you can donate, which is the skin cell, and then also stem cells. Also, we can use preclinical models to assess variants. Some of you might have a VUS, or variant of uncertain significance, on your genetic report, meaning that we don't know whether the variant that your child is carrying is pathogenic or not. I mean, we kind of do, but there's no scientific evidence. And in order to have access to treatment and to a lot of things, you need an actual pathogenic variant for your NARS1 diagnosis. So that is also something you can do in animal models. And last one, I mean, there's many others, but also predict variant mechanism, right? For example, we talked this morning about how having two inherited variants or one de novo variant could give you access to different treatments. And that's something that we're able to study in cells and in the plasma and with functional experiments, and that we can do in preclinical models. So there's different models to study diseases in the lab, and we'll hear about multiple of them this afternoon. Just to go back to our cookbook allegory, so we go from DNA to cells and tissues, and here to living organisms. So we have a human here, we have a mouse, a zebrafish, in a fly. We're actually also studying things in fly. And just to show you different levels of things, I put together well, on my machine. So for example, we can study protein virtually. So that's actually NARS1 right here. And when I said that it falls into this functional bowl, that's what it looks like. So we can look at it and look at the, you have in yellow here, you have the R534 deletion actually. And that is the most simple way to look at it, which is in the computer, less invasive. And then I put pictures of myself because in the lab, for the researchers, please send me pictures of you because that's all I have, pictures of myself doing work. But this is me making proteins in the lab, so making NARS1. This is actually NARS1 in yeast, sorry, bacteria, so using E. coli. And then this is me studying cells, but cells are significantly harder because, as you can see here, I'm working in a hood, so I'm needs to be in a sterile environment. So, and it gets harder and harder as the organism you're working with is alive, right? So, and then the mouse is like the last more intensive work because you're actually working with a living organism. So why do we use neurons, mice, and fly? Well, they're actually very similar to humans. And I put together this little statistics. So percent identity is like the percentage of how similar the DNA is or protein is. So if you look at just all genes in general, well, these are human neurons, so they're going to be 100% humans, but mice with every single gene, like the 3 billion base pairs, they're about 80%, 85% similar to humans, which is pretty high already. Drosophila is, so the fly is 60%, but when you, so that's for all proteins, because obviously they're quite different. So they're non-essential proteins, so they're non-NARS1 proteins are different, but when you go to the essential proteins like NARS1, they're essential for life and they have been for a long, long time actually. Now you can see that they're almost identical in mice and that they're way less similar in flies. And when you look at the variants that we see that are disease-causing in NARS1 right here, they're actually all identical in mice, in neurons, and flies, so which makes them a great model to study just NARS1 in general and those particular NARS1 variants. And when you look at NARS1 symptoms, which is resources that we have on our website, which are all the symptoms that are to this day associated to NARS1, you can see that most of these can be studied in either neuron, mice, or Drosophila, actually. And our researcher will tell you about all the things that you can study in mice and Drosophila. I mean, in fly, you can actually study seizures. You can induce seizures in them. You can look at walking patterns and all those very intricate things. And then, yeah, unless each model is going to have different applications. So human cells, of course, are the closest to what a human is. And in our case, we're lucky that you guys donated your cells to, and we can use actual patient cells with NARS1 variants. So it's human-specific biology, and it's based for molecular mechanisms. So the more detailed things like what NARS1 does in the cells, like more molecular-oriented questions. With mice, we can look at whole organism physiology. So mice have a working brain, just like flies, actually working brains, and we can look at how they walk. We can look at how they behave, whether they're anxious, whether they're communicating with their peers. So there's a lot we can do with mice. And yeah, and they're mammals just like us, so that also makes it easier, but you can also look at EEGs. And for putting drugs on the market, a lot of the FDA requires usually a mouse model in order to get tested with the different therapeutic options. And so we're very glad that we're able to partner with JAX to get a mouse model. And the flies also have a big advantage of being fast and cheap, so they reproduce very easily, but they're still living organisms. And yeah, you can do high throughput screening, so there's a lot of things you can look into. And then, yeah, as I said, you can look for seizures, you can look for slip rhythm, peripheral neuropathies, like CMTs, a lot of things in flies. And yeah, now we can just move on to Jenica's presentation, which is going to be about nursery. Oh, sorry. Any questions about? I was trying to catch up. Any questions about preclinical models? No questions. Do I need to do anything? Do I need to stop presenting? Okay. Yep.
The R designations that were listed are for, is that a variant?
For which one? Oh, yeah, the different NAR1, different NARS1 variants. Yeah, they're disease causing. Yeah.
Okay.
Yeah.